This is a macromolecule lesson, and I wanted to just review some of the macromolecules, actually all of them, C, L, P, and N. If you remember, the C is for carbohydrate, and carbohydrates are a short-term energy source for living things, and they also serve a structural role in the cell membrane. If you remember, in the cell membrane cross-section that you guys had on that last test, those little like flags that were hanging off of the edge of the cell membrane serve as like messengers, and so that's another role. There's also the L, which are the lipids, and the lipids are fats. It's another name for fats, and the lipids are made up of fatty acids and glycerol, and they pretty much are a long-term energy. So that's one thing that we need to remember, is that carbohydrates are short-term energy, lipids are long-term energy. And then P are the proteins, which we're going to focus in on in this lesson. So examples of proteins are enzymes, and so we really focused, out of all the macromolecules the past 2 weeks, we focused on enzymes, which are a protein. So we'll focus on that, and proteins are made up of amino acids, and so those amino acids are many different kinds. There's 20 different amino acids, and the human body has the ability to use a few of those amino acids and arrange them in certain ways to make different types of proteins. And so we'll go over that today too. And then the last one are the nucleic acids for the N, and the base unit of that is the nucleotide, and some examples are DNA and RNA. And then we'll focus in on enzymes and how they function and their rates of reaction regarding chemical reactions and their influence, okay? So that's just a little brief intro. So again, the C stands for carbohydrate. And what we need to remember about carbohydrates is that their monomer or building block is a monosaccharide, which is a simple sugar. And so carbohydrates, like I said before, are a short-term energy source. Sorry, that's an energy, E. And then also I mentioned that the carbohydrates serve as like messengers in the cell membrane too. So they are embedded in the cell membrane, and that helps with the structure of the cell membrane too. And all of these macromolecules are considered not only macromolecules, but also what they call polymers, because they're chains of things. So that's something we should write down in case you come across that word polymer on the EOC in the end of April. These are fairly large molecules that are chain-like and repeated, and so that's another word for you, okay? The L, again, stands for lipid, and lipid is another name for fats, and their monomer is a fatty acid or fatty acids and glycerol. So these are molecules that combine together in a variety of ways to form different types of lipids. And lipids are long-term energy. They're also a good insulator, we've talked about. And they also have a structural role. If you remember, in the phospholipid, right? Here's a phospholipid. The phospholipid, there's two of them because they make up a phospholipid bilayer, bi meaning two. And this tail part is the lipid part. Okay, and you remember the lipid part is also hydrophobic. So it doesn't like water. Okay, so it creates this kind of like layer that divides the internal parts of the cell from the external environment. Okay, and then we have the P for protein. And protein's monomer or building block are amino acids. Okay, and these amino acids combine together in a variety of ways to make different kinds of protein. And they're really important proteins regarding cell processes. So like the transfer of things into and out of the cell. So if you remember, there was the carrier protein and the channel protein. You remember in the cell membrane, because we got to connect all of this. Here's our phospholipids, which are all part of the cell membrane. And you guys should be drawing this, okay? And there was the channel protein. It kind of looked like this in that cross section that we had to know. And so those proteins that are embedded in the cell membrane are very important with transport of things across the cell membrane, going into the cell and out of the cell. And there was also another protein that kind of looked like the channel protein, but it had like a little triangle in it like that. And this was a carrier protein. Okay, and then the cell membrane continues. So it's really important for transport. Also, proteins are important for chemical reactions. Okay, so they're also important for chemical reactions because they help. Specifically, the type of protein we're talking about are enzymes. They definitely help with speeding up chemical reactions in our body because our body's constantly reacting. There's many different types of chemical reactions going on simultaneously. There's reactions going on in some parts of your body and not in other parts of your body. And so there's almost chaos, but it's organized chaos. And it's done so with an efficient means of energy use because of enzymes. Okay, they're super important for all of those things. And then the N are the nucleic acids. And the monomer or building block of nucleic acids are nucleotides. Okay, and nucleotides are made up of a phosphate group, I'm just going to put a P, a sugar group, I'll put an S, and a nitrogen base. Okay, so these nucleotides are repeated units, and we find them in our two examples of nucleic acid, which are DNA and RNA. And a couple of the responsibilities or roles or functions of nucleic acids is definitely hereditary or genetic information. And if you think about it, when we talk about the protein pathway, so this is going back to early September, in order for proteins to be made in the cell, there's DNA, right, that's inside of the nucleus. So the DNA, we're doing the protein pathway here. The DNA is inside the nucleus, right, and it can't leave. Okay, it cannot leave, it cannot leave the nucleus. So it has a friend called RNA, right? And that RNA goes into the nucleus, and it goes into the nucleus through the pores, because the structure of the nucleus, there's little holes in it, and the RNA goes in, and it says, hey DNA, what's your message? And then it gets that message, and that RNA leaves the nucleus with that message. And then it goes to the rough ER. So the endoplasmic reticulum, it'll take that message and it will convert or translate that message into a protein. So then it'll go there and it'll get translated, kind of like translating language. And it'll get translated into a protein. Now what's kind of interesting here is that when it makes this protein, it doesn't just make a protein. It actually has to assemble. It has to assemble amino acids to make that protein, because amino acids make up proteins, right? So it's pretty like all intertwined, right? We've learned that proteins are a macromolecule. We've learned that the monomer or building block of proteins are amino acids. So if we look up here, right, here's protein, here's the amino acids, that's their building block. And we also learned about the protein pathway back in early September. Well, if you think about it, all of that information comes from like the boss of the cell. And the boss of the cell is the nucleus, but it's DNA that's in the nucleus that is sending all this information out. But again, that DNA can't leave, it can't leave the nucleus. So there's a friend, RNA, goes in through the pores of the nucleus, gets the information, and that's actually called transcription. We'll learn soon here in a few weeks. Come on. Again, it's called transcription. Sorry about that. Transcription. And so the RNA transcribes the DNA, leaves the nucleus, goes to the rough ER, right? Goes to the rough ER, and then amino acids are brought over to translate that, okay, and then into a protein. So everything's intertwined, and it's really important for us to obviously listen to these lessons, but also listen to them over and over again and study your notes every day, okay? So out of all of these, we've talked about carbohydrates, we've talked about lipids. These are all macromolecules. We've talked about proteins. We've talked about nucleic acids. And another thing that we've talked about here are the fact that all of them have building blocks, right? And those building blocks are called monomers. Every single one of these has different monomer. Again, the carbohydrates have monosaccharides, the lipids have fatty acids and glycerol, proteins have amino acids, and then nucleic acids have nucleotides. So these are all building blocks, or like Lego blocks, right, to make them the large molecule that they are. So out of all of these, we're going to focus in on proteins. Pictured here is a GIF of a protein, and I want you to notice all of the intermingled, somewhat organized chaos that you see there. Proteins are like that. All of these things are amino acids that are intertwined, and they're almost like kind of like an assemblage of many amino acids, many different lengths, bonded together, accordion-like, okay, like this. And so those kind of squeeze together, right, and they make these long chains of proteins. And one specific protein, well, actually a type of protein that we're going to focus on, okay? A type of protein that we're going to focus on are enzymes. We've been working with enzymes for a while now, okay, for about over a week. And so what we need to know is that proteins or enzymes have some restrictions. And we did that one lab with apples with enzymatic browning. Basically, there's an enzyme that's in apple flesh that, when exposed to oxygen, causes the oxidation of the flesh, which turns it brown. And so in that lab, we were kind of like representing a company that was trying to minimize brownness, because it doesn't look great when you try to sell it. Nobody will buy it. So you want to make sure things don't brown. And so we subjected the apples to different solutions to see what could stop it the best, right? So what we need to know is that enzymes only function within a certain range. And this goes for cells too, because cells have enzymes in them, right? So there's different cells throughout your body that have different requirements. So the cells that are in your intestines, they have a totally different requirement of pH and temperature than, let's say, a cell in your bicep, which is this muscle here, okay? So please understand that. Like, this isn't like all enzymes are the same, all proteins are the same, all cells are the same. They're not. They all have specific requirements. And so they function within a range, a narrow range of pH and temperature, okay? And if the temperature is too cold, then the reaction rate, and I'll just put Rx for short, the reaction rate slow down. Sorry, it's so close together. But then, if it's too hot, I don't know why this is being so weird. If it's too hot, then what happens is the structure of the enzyme is changed. And what we call this is it denatures. It denatures. So all of those times that we drew, like let's say this is a substrate and then this is the enzyme, and it would like fit perfectly in there. And then just to recall, when all of that is together, that's the enzyme substrate complex. And then when that enzyme is done working on the substrate, it'll produce some products, and then that enzyme will be free to be reused again, right? Well, when, let's say that this enzyme is exposed to really hot conditions, then that enzyme will no longer look like that. It might look like this, okay? And so then that substrate does not fit inside of that enzyme because now it has been denatured. It's been denatured because of extreme conditions, specifically in this example, the heat, okay? And so another thing we need to remember is that not only do they have this range within which they work, but we also need to know like how awesome they are. And that is the one reason is because it lowers the activation energy needed for reactions to occur, okay? So proteins, or specifically enzymes in this case, lower that activation energy. So if you had a graph and you had the amount of energy needed and then the reaction rate or time, let's say without enzymes, that activation energy is this hump here. That's how much it costs for a reaction to occur over time. If you were to add an enzyme to this, this is what would happen. So you have your reaction rate going, and then you add the enzyme, and that little hump there is lowered. Okay? And so since that little hump is lowered, it reduces that much energy that's needed for that reaction to occur. Okay? So right there, that's your savings in energy. And so I think that's it. So we just need to remember the whole activation energy thing and the fact that enzymes lower it, and as a result, what this does then is it speeds up reaction rates. Okay, super important. All right, I think that's it. Good luck studying for the test. I think you guys are going to do great. Just remember to study, look at that review sheet, study it, look at your notes that we've been taking in that macromolecule packet, and you'll be good to go. All right, see you later.